This is episode 87 of the Immunology Podcast, Applications of Immunology with Dr. Juang. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Juang from STEMCELL Technologies on the podcast to talk about his work as a field application scientist in industry. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
Immunology 2025 is the world-leading annual all-immunology meeting, and this year is in Honolulu, Hawaii. Join us from May 3 to 7 of 2025 to connect, collaborate, advance your career, and access cutting-edge immunology research. And of course, enjoy Hawaii. Abstracts open for submission on October 1st, 2024. So that's around the corner. Learn more at immunology2025.aai.org.
So Brenda, our guest this last time around went back to school for an MBA, right?
He did.
Would you ever go back to school?
I would say if just going to university was a career and you could make a living out of that, I probably would be open to the idea. I'm not sure if I would do an MBA though. I would train as, I don't know, as a pilot or something like completely different, something very active and a little bit adrenaline inducing. That sounds kind of fun. An astronaut.
All right.
You?
No. I've done 32 years of school.
Yeah. You had enough, didn't you?
Literally, I will be in almost 70 before, 64, when it'll be half my life out of school. So talk to me then at episode like 500 or 1,000 or whatever we'll be at. And then, or even further along, and then we can decide if I'd go back to school. Then I'd be ready. Then I'd go back to culinary school. So when I retire, I would I would go learn to cook or something for a year or two, but like in my 60s.
That sounds kind of nice. You could take one of those, trips to Italy, and then you learned how to do pasta. Well,
yeah, like retirement vibe, like educational plan here, right? Like
I love it.
A course here or there, no grades. Pass, fail, just acquiring knowledge. I do that. But like if it's school, I'm cooked.
Okay, I understand. It does take a toll. It does take a toll on the soul. So much schooling. All right. Talking about schooling, what are you going to school me about today, immunology-wise?
Tuberculosis.
Ooh, all right.
All right. So this is in Nature. It published 28th of August. Tuberculosis in otherwise healthy adults with inherited TNF deficiency. The notations on this paper are weird in terms of authors. So I'm going to say the first author is Andre A. Arias. Corresponding authors, which are all over the author order, include Ana Lena Nihas, Jose Luis Franco, and Jean Lorette Casanova. And the last author is Stephanie Boyson Dupois. So if I missed anyone or anything, I'm sorry. This is what I can gather from the online publication. I'm trying to do it right here for the shout outs. All right. So this is it's a quick story, but a really cool story in that we know most people who get are really susceptible to TB genetically have been identified as having problems with interferon signaling. And so there's work looking at people who are susceptible to tuberculosis or other mycoplasma and what that means and understanding mechanism from that and just, natural genetic mutations that arise in people. Okay. Well, so there's sentinel programs for this to understand how to work on TB. And this family, which is someone and their first cousin, popped up, but they didn't have anything wrong with their interferon. They had a private mutation that affects the TNF gene and truncates it and makes it useless. And these two patients got bad TB over and over again. So why is this paper cool? I'm going to tell you the why, and I'll talk a little about what they showed through. So why is it cool? It shows a whole new pathway of how important it is for TNF to mitigate tuberculosis function. So TNF is a required molecule. Now we know that people on anti-TNFs are more susceptible to TB, but this is the first time it's like critical front and center, single gene, bam, you get TB all the time. So what did they do? They identified these people, they took their blood, they figured out what this mutation was, right? They sequenced them, figured out the mutation, showed that TNF was non-functional, did induced macrophages, and looked at what was happening and looked at comparisons to TNF or TNFR1 deficient stem cells in comparison, right? So they made their other lines that they could then imitate with, right, other mutations, and showed that it was TNF through the TNFR1 receptor, not the TNF2 receptor, but just the single TNF receptor that is super critical for this all to function. And it is a vital part of the pathway. And so then they also showed that it affects Bacillus Calmette-Guerin syndrome in addition to TB. So this is that contrasts with that, who are prone to multiple infections. It's really just a TB thing, and it affects the ability of the macrophages to induce a respiratory burst. Right, so they go into the cells, they identify that the TNFR1 receptor through TNF is engaging the macrophages to do a respiratory burst that is lost in these cells from primary patients or in cell lines they make that mimic this, right? So they can do hex cells that don't make a lot of TNF and then put in regular gene or the mutation or what have you. They can take the mutation, introduce TNF into the culture, and it still works. So long and short, it's the TNF that induces an oxidative burst in the macrophages. That's what controls macrophage function. And they look at the sequencing and other stuff, and they don't find huge disruptions in cell types. There's some lower numbers of some cells, but nothing wild. Leukocyte development is normal. They look normal except for this loss of function largely, or slightly perturbed, but not anything that like is a smoking gun of a loss of function from development or just none of one type of cell or wild genetic differences at baseline. They'll see different gene activation in the knockout, right? You don't have NF-kappa B driven TNF activation in the mutation, right? But at baseline, at rest, it's shockingly unexciting. Except when you push the system and they have to do an oxidative burst, and then it doesn't work. So it's low reactive oxygen species once you push them.
How interesting. So these people are related, but then the mutation is the same mutation. I didn't understand.
It's the same mutation in both of them. They are related individuals. It's a private mutation, so it's not one of those publicly known mutations that exist in the scientific ether.
And when it comes to other, do they do any other studies with other infectious or maybe other stimuli? Would this mutation TNF receptor also affect susceptibility to other diseases?
Or as a sex susceptibility to listeria.
Hmm. Okay, there's something that can do in vitro, I guess.
Yeah, in vitro, right, with another oxidative burst dependent system.
But these patients didn't report susceptibility to any other specific infection, just the TB that
Correct.
caught their attention. Huh. It's very interesting when you have these kind of human cases that allow you to study like these natural experiments.
Yeah.
Are very, very cool. I mean, I feel sorry for the patients, of course, but you do get to learn a lot.
Right, and they could get recombinant TNF therapy or something. I don't know if
Yeah.
these people can. That'd be very expensive. I don't even think that exists in the market, but like it lets you know some things, right?
Yeah.
There's a path forward now.
It's interesting because also, given if they are from Colombia, what is the age of these patients?
18, they first had TB. So they started developing TB at 18 and 19, and then they're in their 20s and 30s.
Because I guess that if they're from South America, around that age, they're probably double vaccinated with BCG, but I guess it doesn't really help against pulmonary TB at all, or does it? So
I think that's probably why they waited so long to present, but then eventually they can't fight it off and they get it. And then they cleared it and got it again, which is really weird.
Right. Yeah.
Yep.
But they got it with treatment, with antibiotics.
Yeah, they killed it with antibiotics and got it again pretty quickly. They're like, wait a minute, you're someone that
Mm.
we got to go screen for genetics. And so that's
All right. Very interesting. Thanks for sharing.
all right. Brenda, I gave you a little bit of TB. What are you giving me in return?
I'll just give you some inflammation and neuropathology in COVID-19.
Oh,
How
so
about
you
that?
give me the Rona.
Yeah. Yeah.
All right.
Well, so or not, or maybe I'll give you a solution for it. See how this turns very, let me just put a hopeful touch to this. All right, so the paper I want to talk about is called "Fibrin Drives Thromboinflammation and Neuropathy in COVID-19," published in Nature on the 28th of August, and it has a bunch of first authors. So we have J.Q. Ryu, Sheiko Yan, Mauricio Montano, Elif Sussman, Karuna Dixit, and Rahul Suryawanshi from I guess the a collaboration from the corresponding authors Warner Green and Katerina Akasaglu from Gladstone Institute and the University of California, San Francisco. So, as the title suggests, this paper talks about inflammation and in the context of COVID, and tries to connect, the very well-known phenomena of clotting and coagulopathy, which occurs in patients with COVID, fairly frequently, and also particularly in young patients, and also sometimes associated to mild infections, and that this, to some extent, is associated with the neurological complications and that are present in COVID. So I have to say, this paper, I liked it, but I saw some kind of, I don't want to say criticism, but some kind of comments on Twitter about it. And I think I kind of agree. I think it's um focuses a lot on this connection between clotting, neuropathy, and it tries to make a connection to long COVID because we know that this, the brain fog and the neurological complications are part of the long COVID kind of syndrome. And they try to show that, these things are related, directly. They're kind of a causal relationship. So one of the things that they indicate is that you have these blood clots in COVID that can persist despite anticoagulation treatment. So there's something about these clots that is kind of different. And I think this research line comes from the understanding that fibrin, which is derived from the soluble blood protein fibrinogen that gets activated upon coagulation and forms this now the main component of a blood clot. And they had, I think, other previous research talking about, I think, fibrogen and I find fibrin in other diseases. And I think they really want to highlight a particular antibody that can prevent kind of uh, the some of the complications that come from clotting that have to do with inflammation caused by clotting. So they want to look into clotting and fibrin and how this relates to SARS-CoV-2, and they show that fibrinogen can bind to the spike protein of SARS-CoV-2. But this is the first connection they make. They make some, in vitro assays, some solid phase binding assays, and they do some, they show that there's binding of fibrinogen and fibrin to spike. And if they have some mouse models that show that uh, there are colocalization of these proteins in infected mice, and they do a lot of kind of more like biochemical mutational experiments, and they show that there's specific binding sites, and there is one in particular that seems to be the major binding site between spike and fibrinogen or fibrin, that is a specific spot, and this spot uh, coincides with a cryptic fibrinogen binding site for complement receptor, and it's a part of fibrin that seems to be associated with activating um, macrophages and myeloid cells to generate ROS and activation and cytokines and things like that. So basically they suggest that spike can bind to fibrin and kind of activate they generate these clots that have this activate this site that really activates can activate myeloid cells. So they have these mice, or they have mice that are lacking fibrinogen, and they show that absence of fibrinogen or mutants that lack that motif that is kind of interacts with myeloid cells are don't have these fiber deposits that are observed in mice infected with SARS-CoV-2, and they have less inflammation, oxidative stress. And also very interestingly is that they see improved activation of NK cells in the lungs of these mice infected. So it really seems to affect the the immune response. So this clotting seems to be this is fibrin clots affect the immune response on the one hand by reducing NK cell recruitment, and this of course has negative effects on virus clearance, but then also by activating macrophages. Um, and and so they show that um, they have this antibody called 5B8 that can target this inflammatory domain of fibrin. And they show that by doing either prophylactic administration before infection, which I guess is not so interesting, but they can reduce macrophage activation, oxidative stress, collagen accumulation, and fibrin deposition um, without really changing viral titer, but they can reduce kind of the inflammation. And some of these decreased inflammation can also be obtained by therapeutic administration 24 hours after infection. So it also seems to help a little bit after. So what is interesting, and I think kind of the point they want to make is that similarly, they this fibrin clots or this fibrin deposition seems to also affect microglial cells in the brain. And this is both in condition in mouse models with viruses that are neuroinvasive and not. So even if the virus is not replicating in neurons or in the brain, you still have deposition of fibrin, and you still have activation of the microglia in the brain. And this can be reduced by not having fibrin. So by having these mouse models without fibrin or with this missing domain, or to some extent by using this therapeutic antibody. And this also is in the case for neuroinvasive models. So basically, the connection they want to make. So they show they had a model in which they have expression of spike without infection, and this also seems to correlate with fibrin deposition. So they want to say that if you have spike, that seems to be sufficient to generate these clots that are inflammatory, and this might be mediating things like long COVID. They don't have long-term models in this experiment, so it is a kind of hypothesis. It's kind of a injecture, but this that's kind of the point they want to make. And of course, you have this, antibody that targets this fibrin, this malignant fibrin, and it wouldn't be great if it could have therapeutic uses in human and particularly in long COVID, which has grown to be such a huge problem nowadays.
So you had me for the first part with everything up to long COVID, and especially the
Mhm.
notion that spike is the cause, because we see some of the clotting things even with the vaccines, right? Which is all spike often,
Yeah,
right?
they push back against the idea that the vaccines themselves are there's enough spike made by the vaccine. I
Uh,
don't know. They don't talk about it. I mean, they have no
I mean,
vaccination models here,
J and
but
J's drug was pulled for clotting from
yeah,
the
but that's a different, and they make that point in the paper. That's a different reason. Like this clotting, because they have this activation. It's a different mechanism because people have already kind of established that the what they observed in the Johnson vaccines is what was it? This PFA4. What
kind of, but
was
there's
it?
an adenoviral component, and they think it has to do with adenovirus and spike causing clot.
Yeah. Yeah, so I mean, it is true. There's no experience here to show either way, but there is a mechanism for that clotting that has been identified that is not this one, or they could be complementary, I guess.
Yeah. I think the other thing is I don't know why they would say you have spike forever and ever with long COVID, because long COVID persists months after you have no virus left. So why do you have long COVID? Because the fibrinogen persists for months and months and months.
Well, apparently this binding to spike does make this fibrin more resistant to lysis. So it could uh kind of make these clots last longer than they should.
Yeah, but you're
Um
talking months. I don't know. I don't buy that one.
yeah, I know.
I'm going to call stretch here because that protein should degrade in months, maybe 2 months, not 1 month, but that's still not long.
Yeah. Well, I don't know. There are some report. I had to admit I'm not completely updated, but I thought I heard that were some reports that where spike could be not the mRNA, like the actual protein could be detected many months after infection. Although I guess it must be really hard to, be sure when the last infection was. So I don't know. Yeah, that's the thing. I think the connection with long COVID is a little bit of a stretch. I still think it's exciting. Um, but yeah, I don't think so. There was a lot of like hype around this. I saw some headlines about it, but and they like literally the first sentence in this paper is long COVID is a big problem of health in public health. It's like, okay, so you're hearing, but well, I think also to some extent this paper wants to highlight this antibody as a therapeutic antibody. They obviously have patents for this antibody. So yeah, I mean there might be a little bit of a um
Push.
yeah, yeah. I mean,
Understandable, but just calling it out. All right.
yeah.
So you gave me COVID. I'm going to give
Okay.
you sodium chloride.
Awesome. Just can
I'm
put some
going to, I'm
salt
gonna,
on my food.
and cure you of cancer.
Oh, wow, I love that.
Like,
Okay.
if we're being hyperbolic here. All right. So this is Nature Immunology, also the 28th of August. Sodium chloride in the tumor microenvironment enhances T cell metabolic fitness and cytotoxicity. First author is Dominic Soll. Last author is Christina Zielinski. All right. So, in the pile of things I didn't know about till reading this paper, apparently the salinity of the tissue near, or the extracellular tissue near cancer cells is higher than normal. And that matters. Okay. So, they have some background about why this was the case. I don't know how they actually got to going down this path, because I think sometimes you write the paper after you find the interesting thing and make a coherent story, but it doesn't matter because it's cool. So they show that they determine the concentrations of sodium and potassium in intratumoral and peritumoral tissue, and intratumoral is higher. And they do this with inductively coupled plasma optic emission spectroscopy, IPC OES, which, based on the name, you're going to blast something, turn it into plasma, and then measure its light emission to tell you how much of various metals like potassium and sodium are in there on pieces of tissue, I think is what they do.
Like, well, like we used to do that in organic chemistry. Just put some sodium on your burner, and then you get a nice color. Anyway, continue.
Yes. Boom. All right. So there's more there. They then established a transcriptomic gene signature of sodium chloride exposed CD8 cells by taking CD8 positive CD45 RA minus T cells with culture with CD3 and CD20 monoclonal antibodies under low and high extracellular sodium chloride conditions. They did this culturing and then did an RNA-seq to determine a signature. They made that signature and describe it. And there's genes that go up. And then they found that in an atlas of 30, it has had a whole total of 14,000 samples from 9,000 different patients and 25 distinct tumor types from publicly available Genome Atlas and GTEx projects, looked at it and found that in almost all in breast cancer and all other solid tumors, except tumors of the thymus and paraganglia, there was an enrichment in the sodium chloride signature relative to paired healthy tissue. So, hey, they found this pattern and it appears in existing data sets. Cool. That tells you you're onto something. They then looked at public available sequencing from CD8 positive tumor infiltrating lymphocytes and saw that too. Right. So they went to another data set that was that subset and found it. So what does it do? So they spend a lot of time now in the paper figuring out what it does. It increases T cell activation and TCR signaling in the CD8 positive cells. That's the first thing they establish it does, using basically, human acquired cells that they then induce and push the system on. Right. They do single cell RNA-seq. They look at all the things. They look at how the TCR, so TCR stimulates calcium influx, and they find that the sodium increases the membrane polarization in such a way that promotes calcium influx afterwards. So it primes this. And that then potentiates cytotoxic T cell effector function. And they show that, when you add, TNF expressions induced, IL-2 expressions induced, other genes and activities induced, degranulation signaling is induced by this. Okay. They then find, well, what else is going on? Enhances metabolic fitness because with this extra sodium on the outside, you're now influxing glucose and other things, and that makes them have this extra metabolic fitness to them. They're burning more oxygen, they're burning more glucose. They see both glycolysis and oxidative phosphorylation increase. We know extracellular sodium influences mitochondrial function as well, and they're seeing all this change. And there's changes in the lipid metabolism is uptaked, and there's more glutamine to glutamate ratio is enhanced, which is another thing apparently affects cytotoxic T cell differentiation. So they're saying that there's hyperpolarization. Oh, and they also identify that due to the sodium potassium ATPase. Duh. Right. So the sodium potassium ATPase is responsible for the downstream changes from having extracellular sodium, which then influences calcium influx, which means that you have a stronger signaling burst when you do signaling on the cells. And this also extracellular sodium makes the cells uptake more nutrients and be more metabolically fit. And then if you take these prime cells and do it and inject them into tumor models in mice or put them co-culture with human tumor cells, they kill better. But guess what? So that's okay. That's cool. Like, okay, this is cool. Where's the final money? They then add this to a CAR T cell regimen to make your CAR T cells, right? So how if I make my CAR T cells, what if I give them some extra sodium and the cells work better? And that's the final bit. Like, so the real point of this is they figure it out, but in the end, it's like, yo, we should put some increase the culture condition sodium concentration past normal by about 35% because it makes the cells work better, and it makes them work better even when you put them in a regular environment afterwards. The changes persist, at least through therapeutic window. Boom. That's the cool part.
That's so nice. You just add a little bit of salt to your cells and
a little salt to the wound really hits that cancer hard.
makes them taste better. Work better. Okay, that's very interesting. Well, I seem to remember like the story about salt being bad for immunity. I don't know. I have this idea in my mind, but maybe I don't know which was the context. But this is like cells ex vivo, like in vitro. It's not like you can just start eating, more salt
Yeah,
if
that's
you
going
have
to be really
cancer.
complicated. They see that, and they also see that a high sodium signature is indicative of better outcomes. So for that signature in tumors, so that people live longer if they have a high sodium signature.
Huh.
So there's something there, and it seems to prime immune cells, but but your body's going to do everything it can to maintain stuff.
Yeah, it's not like you can change that, really.
But you could condition your CAR T cells the right way and make them even better.
All right. Very interesting. Okay. To finish up today, I will stick to, respiratory infections, and I'm going to give you some genetic or, predispositions to severe disease and the reasons behind it. So this paper I want to talk about is called High Expression of Oleoyl ACP Hydrolase Underpins Life-Threatening Respiratory Viral Diseases, published in Cell 22nd of August. First authors Xiao Jia and Jeremy Chase Crawford, from corresponding author Catherine Kaczyrska from the Peter Doherty Institute in Melbourne, Australia. And I want to make it shorter because I think it's a very nice, sweet, and short and sweet story. They start with an observation. They compared. They had transcriptomic data from patients that were hospitalized during a fatal influenza season, this H7N9 in China, and there's also this avian influenza that had a very high mortality rate. And so they kind of started studying, samples from patients that survive and patients that died, and trying to understand, the components, immunologic component behind this difference. And so, you know, old school, they started I'm sorry, some people are gonna hate me for saying this. They started the story with results from a gene array, an F matrix gene array from samples from these patients, and they show. And so the observation was that this enzyme, oleoyl acyl carrier protein hydrolase, uh, called OLA, was one of kind of the main differences, one of the most kind of surprising differences in expression levels between patients that died and lived after H7N9 infection. And so they really show that there's a lot of data and different conditions, and then they show that they study mRNA levels. They show that it is expressed predominantly on CD14 monocytes. They see that this difference in expression of OLA is also observed in other conditions, in other ICU patients that have influenza. They also have elevated expression of this enzyme. And in patients with COVID, those that have severe disease, they show kind of a peak expression. They start expressing early after infection on day 5, they peak on day 9, and they keep this expression throughout the length of their severe pathology. And this is not the case for patients with minimal disease. And so they were kind of pinpoint this OLA enzyme as being kind of important in this severe presentation. And so they have OLA knockout mice, and they show that these mice are protected from severe influenza. And they show reduced pro-inflammatory cytokines in lungs, in bronchoalveolar lavage. They have reduced macrophages, neutrophils, and other inflammatory cells, and NK cells. And they show that this protection from severe influenza depends on expression of OLA or absence in the bone marrow. So they have bone marrow chimeras, and they show again it is the expression of OLA in the bone marrow and the kind of hematopoietic compartment that makes the difference. And so they show that there's this production of lipid droplets they see in infected lungs, and they kind of zero down a little bit on macrophages and how this macrophage is really activated as well. Oleic acid is the main product from OLA enzymatic activity. So, and they see that also they can to some extent increase the severity of influenza in mouse models by increasing the dietary intake of oleic acid. So they show basically what they conclude is that these these oleic acid increases the activation of these macrophages in severe disease, and so they have more pro-inflammatory cytokines, and this affects the severe disease. And I think when they talk about kind of the mechanism, they show that these lipid droplets that are found in the lungs, they support uh, viral transmission, or like viral replication. So these are part of the pathology. And also that they make the connection with oleic acid, uh, kind of impairing some of the antiviral mechanisms in macrophages. And therefore, they talk about these IFTM3 in particular, seems to be inhibited by oleic acid and contributed to the increased infection observed in macrophages that are expressing high amounts of OLA. So the high amount of this protein uh, exposes you to severe disease, and they think that you can even use this as kind of a biomarker to expect, because it's already early on an infection, so you might already be able to tell whether a patient might be, heading towards a severe disease, be by measuring oleic expression and oleic acid as well.
Hmm. So this is just how some people are?
That was not clear to me. Yeah, I don't know where the variability comes from. So it is not clear to me if it's like these people are already from the baseline expressing more Ola. I don't think I got that from the text, honestly. Because we know, right? There are other examples of polymorphisms or mutations or expression like that will affect susceptibility to infection. But I don't know if this is the case. How exactly is Ola regulated and the levels of expression regulated? I don't think they know either. For the
It's just so it's observational.
Yeah, yeah. So they provide some kind of mechanism of why having this moralic acid seems to increase infection of and macrophage inflammation, and that how this oleic acid interferes with some um, interferon uh, induced genes like IFITM3, which would protect to some extent macrophages and initiate the antiviral response. So, but it's not clear where the variability is coming from.
Interesting. Hmm. All right. Well, I may know where I'm coming from, but not where I'm going at this point. But I know what I'm doing next in life, at least. And that is that we're going to have a great interview here with Dr. Wayne Juang from STEMCELL in just a moment. But before we get to that, explore scientific resources for your immunology research at the STEMCELL Technologies Immunology Learning Center. Choose from different research areas and find expert interviews, technical tips, educational webinars, instructional videos, and much more. Visit stemcell.com/immunology-research.
Now, for the second part of today's episode, we are very happy to be joined by Dr. Wayne Huang. He is a field application scientist in immunology at STEMCELL Technologies, and he's going to talk to us about her role at STEMCELL, her experience at industry, maybe some advice for those considering making the move. So I'm very happy to have him on. Thank you so much for joining us. Welcome to the Immunology Podcast.
Thanks, Brenda. Thanks, Jason, for having me on here. Pretty honored to be on the show and talk about my experiences.
Thank you for being here. All right. So you're a stem cell field application scientist. So I'm going to start with the obvious, easy question, which is what the heck is that?
Yeah, that's a good question. So field application scientist, I mean, when you look at the face of it, the field means that we do a lot of field work, right? So we're traveling a lot, going actually to customer sites, visiting with customers. And then the application scientist part typically refers to, doing demos, right? Talking to customers, doing the science with them, talking about the science with them, right? So in a sense, it's a blend of R&D scientists mixed a little bit with sort of the sales aspect and roles.
And so maybe what does a kind of average day or an average week looks like for you in this position?
Yeah, that's a good question. The day-to-day can be different. Weeks can be quite different, but maybe I'll give you a better picture of what might like an average month look like for me then in that sense. So usually in a month, I might be traveling to 2, maybe 3 customer sites. So I'm based out of Vancouver, right? And that means a lot of our customers are in the States, right? They're in the California area. They might be in, let's say, the Michigan area, Ohio, or even down in Florida. And basically, what I would be doing is when I go on the sites with the customers, it would be about troubleshooting some of the workflows sometimes. I might be demoing some of our products with the customers, right? I mean, customers always like to, I know for me, like test drive the product before you buy it, right? And so really that's what we'd be doing on site. And so those travels would take about, a week or so, a couple of days. And I'd be doing that trips maybe 2 or 3 times a month, right? Now, when I'm out traveling, I'm home based, as you can kind of guess. And during that home based time, I'm working with R&D or some of our other FAS people, and working on materials to help promote our products more, right? Or troubleshooting customer issues through phone calls or virtual meetings.
So, with that, do you usually know what the problem is when you're going out? So you're not going out just for the regular thing. There's a specific problem. Can you give an example, obviously without anything proprietary, of a type of problem that you're sent for?
Yeah. Usually, yeah, we do know the problem. So if it's a problem where a customer has tried, let's say, our kits or have tried something more complicated, right? So a lot of our kits, you can use them in tandem, one after another. Maybe they're trying to isolate, T cells from the same sample as the B cells, and then K cells as well, so that you get basically all the cell types from the same donor, same sample. They may run into issues where the recovery is not as high as they like, right, or they need, or the purity somehow is not as high as well, right? So we kind of know those problems ahead of time. And that's usually where, the weeks where I'm not traveling, I'm working on resolutions with that. A lot of times I'll be talking with the sales rep, understanding sort of what was done in the past, what they're seeing, then also reaching out to R&D and seeing, what do they have, do they have any ideas? What might be the issue and how we can improve sort of what the customer needs. Right? So yeah, it's a large part. I do know the problems ahead of time, but a lot of times too, when we're on site, new problems pop up, right? Or a neighboring lab has an emergency that pops up. And then in those cases, we just kind of go in to do our best to understand the problem and then try to see if we have resolution then or there. And if not, kind of take the problem back to Vancouver and discuss it with my team.
So in a way, it's kind of you're bridging the R&D, sales. So you're there in person with the customer, but then you're also, I guess, in close contact with the people at the R&D and kind of in and out all the variables and all the things that could go wrong, and all the explanations behind each of the products.
Yeah, absolutely. I mean, the bridge analogy is great, and I love that because that's kind of what we do. We sit in between, I think, a lot of three connections, really. You have the R&D on one side, we have the sales team on the other side, and we have the customers, in a third dimension instead of in a way too. And we're in the middle trying to bridge those three together, sort of to be the in-field, in-person sort of scientific expert, and then trying to communicate our scientific knowledge, our scientific expertise to the customer to help them resolve their issues. So it's a really great place to be because you can kind of have, that three-way connections and feel that you're the center linchpin to hold everything together. And I think it's very rewarding in that sense too. Yeah.
This is kind of fun. There's never a dull moment, I guess. Always different problems, always different people. That sounds kind of interesting.
Oh yeah, no, definitely. It's never the same problem twice. It can be very, very similar. A lot of times it sounds like the exact same problem, but then when you actually dig deeper, you realize, there's just a little bit of difference that makes it a whole new set of issues that we had to work with.
So what made you decide to go down this career path? Did you know you always wanted to go into industry? Was this a shift?
Sure. I think I've always thought that I would either probably end up in industry at some point. During my PhD, I love the science. That's why I got into immunology. The complexity of immunology is so wonderful, right? It's just so complicated that there's no one answer for a lot of things. But I've always kind of known that, the academic life may not be the one for me in that sense. But also during my grad school years, I really enjoyed talking to the sales reps that came by. I know that's not a lot of fun for a lot of grad students, postdocs. You're trying to get your paper out, to have someone come ask you questions about what you're doing. But for me, I always enjoyed the communication aspect, and so when I kind of looked at sort of where my career path would go, this was kind of always the direction I wanted to go into. I want to be in a place where I can utilize my scientific knowledge that I've learned, but then have a communication aspect to it. And so and thinking about this position, and that's perfect, right? Because I'm out there talking to customers, understanding their issues, understanding their projects. But at the same time, I get to use my sort of scientific expertise and knowledge and experience to help them push their projects forward. So I think it was a natural fit. It was a really good one as well.
And did you start at STEMCELL, or did you work other places before coming to STEMCELL?
I did work at one place before I came to STEMCELL. So after my PhD, one of the things I thought would help in sort of my experience development aspect was, I had the sort of the academic research experience with the PhD for 6.5 years, that, I wanted to get some industry research experience to kind of see what the difference is, right? And so right after I graduated, I joined a company here in Vancouver. We were doing sort of cancer therapy or cancer treatments. And so I joined on the team as a research associate. Was there for about 3.5 years, right? So it's a good enough time to understand some of the issues that industry research might run into that I didn't necessarily see in my academic life, right? And so it helped me understand a bit more of the troubles that would happen on both sides, right? And then from there, research was fun, was great, but we worked a lot with mice, and I decided it was a time for a change to come into STEMCELL, to be more on the scientific communications aspect, right? And from there, I kind of first joined the product scientific support team. Basically, it's just troubleshooting team where if customers have issues, we would get a phone call, we get an email to help resolve that. And from there, I love that aspect of the role. And then from there, moved into the FAS roles a couple of years later.
So, what was for you? How did you feel the transition? So you said that you already had an idea that you want to work on this. So I guess you were kind of mentally preparing yourself for the transition.
Mm-hmm.
But for those that are not doing their PhD or their postdoc, what was for you the main kind of take home message from that transition from academia to still maybe a research position, but in industry, and that you would like to share maybe with our listeners?
Yeah, so I think the main difference between, so you're talking about what main difference of like academic research and industry research might be.
Mhm.
I think the main difference that I had to cope with or sort of better understand going in was that in academic research, the the goal is to find novel scientific discoveries and publish them, right? Or to get the next grant. But in industry, the goal really is to make a product that is profitable for the company, right? It can be a diagnostic product, it can be a drug, but there is very specifically, it's no longer, oh, if this is interesting, let's pursue it, right? It has to be, if this is interesting, it's going to help the company, but more importantly, it's going to help a patient in some way, right? And so in that sense, your focus is a lot more narrow, and it's much more directed, right? And so that can be kind of a good and bad thing, right? Because it really focuses you on certain things that you have to do or you can't do in some ways, right? But in other aspects, it becomes difficult because as scientists, we're always curious about every single question that we can't answer, but then we can't pursue them all, right? So that was kind of the two sides of the coin that was a little hard to choose, right? Sometimes, and I think that was probably the most difficult thing about that transition for me.
And you also ended up doing an MBA recently, right? Could you talk about the thought behind going for that and what that experience was like?
Absolutely, yeah. I think first and foremost, the one thing for me was curiosity. I was always interested in the business aspects a little bit as well, right? Just to understand a little bit more about our world, because it's not just the science that drives everything, right? It's the science, it's the business, it's the arts, it's the politics. It's all the different areas of study that kind of interconnect together, right? So for me, the more that I can understand the world out there, never really hurts, right? The other aspect of that was after doing research in industry for a while, I did realize there's a lot more of the economic side of things that I really didn't understand, right? The decision makings behind which projects we pursue or not pursue, right, is also driven by a lot of the economics. And so in that sense, coming into this role or knowing that I'll be basically working a lot with our customers, I felt like this is an area where that can help me understand them better, right? To understand sort of their economic backgrounds for the labs or the companies that I'm working with and troubleshooting with, to better understand their pain points, right? To then get them from A to B, from where they are now to ultimately where their goal is going to be. And so that's sort of the thinking behind it. And so that's kind of why I pursued the MBA. As for the experience, I loved it. It was a lot of hard work, a lot of late nights, but it was amazing. It's a how should I say it? Like, it's really an environment where I can interact with a lot of different people from a lot of different fields that I would never have encountered, right? I mean, take our class, for example. We had about a third of our cohort being in finance, a third being engineers, and the rest of the third being mixed between, lawyers, doctors, biotech, such myself, right? So when you look at the whole room, when I look at my classmates, I'm in an environment where I would never encounter 2/3 of these people if I just stayed in my field, right? So I get to see how other people think about the same problem, how they approach it, and I think ultimately that's going to help me better understand our customers, right? How do they think about doing things? It's not necessarily how I would think about it as well. So I think it's a great experience. Despite the tuition, no regrets for sure. And yeah, do it again in a heartbeat if I had to. Yeah.
That's very interesting. Because these are some programs that you can do next to your job, right? You just,
Yes.
it's designed so that you're doing your regular day job and then you can do an MBA on the side. And it's mostly professionals like you that have already several years of experience and they just want to kind of level up in a way.
Yeah, that's right. I mean, the program that I took was sort of the part-time or what the university called the professional MBA program. Basically, our classes were all on weekends. So they would be, Saturday and Sunday, 8 hours a day. It's every other week typically. And so it's quite intense in the sense that you're working 8 hours a week, and then on the weekend you're taking 8-hour classes every day. But no, it's something that we can that works well with sort of the working life. And yeah, everyone in a class, everyone in class also is working, right? So it does help with understanding the different challenges that different professions have as well, right? But no, it's one of those that it's great to have that experience there. Yeah.
So, one more thing about your position and your work, with the customers, with the scientist, maybe more on the kind of just to geek out a little bit about the kind of services that, companies like Stem Cell provide. Is there anything that excites you most about the work? Any kind of particular, you know, maybe a particular protocol assay or like cell type of things that you think I really like when customers come with this kind of problem. This is what excites me the most. I'm like, oh, this is the kind of problems I want to solve in my immunology kind of area of concern. So it's just for our guests or something more like specific for our guests to think about.
Yeah, I mean, one of the things that is one of the products I really love about our company is the RoboStep-S instrument. Automation is wonderful. It saves everybody a lot of time. It's a lot more consistent. I know in grad school, I did a lot of cell isolations when I was in grad school as well. And I wish we had the funding to buy an automated machine, right? Because it makes life so much easier. Not just for the technician or the grad student that's using it, but also it brings a lot more consistency to sort of the isolation experiments we do. And I mean, I know when I had very, very long days, toward the end of the experiments, I'm just guessing on myself, did I add the right cocktail? Did I write the right particle amounts? But with automation, that, the isolation will run exactly the same way every single time. Machine doesn't get tired after, say, 5 or 10 different isolations. So that's one thing, that I always love when customers ask me about automation and sort of the benefits thereof with that. The other one, of course, I always like to tackle is troubleshooting issues, right? Where customer says, "Oh, I've isolated these cells, but I'm not really getting the numbers or the purity that I'm looking for." And I think that's also very rewarding because, in a sense, yes, if we can help fix the issues for customers, they're happy and it's wonderful. But on another note too, the more we can help the customers out, the faster that science itself will progress, right? If we're able to get an extra, say, 500,000 cells each time, that can be an extra well in experiment. That could that builds up over time. I think about triplicates. You get an extra well, you get an extra dosing that you can test. And then over a year, right, that could be an extra compound, maybe 2 or 10 compounds, right, depending on what they're doing. And that just means that we can get, bench side to clinical side a lot faster, right? And so I think those are two things that really excite me when I get requests to come out and visit customers for sure.
So I guess my last question on kind of the whole career thing and everything else for you is, now with an MBA and you've done this, is there like kind of where you want to go next in your career from field application scientist? Do you see like, how does this look long term, right? And especially for not just you, but others.
Yeah, no, that's a good question. That's something I'm continuing to develop, I think. The MBA really allows me, I feel like, has opened a lot of doors, potential doors for me, right? There's a lot of doors now I can walk through. One, of course, is, going higher into management, right? So then I can basically bring more benefits to the customers of the company, right? Because in the MBA, it's not just about the business, the economics, right? We also have a lot of courses about team management. How do you develop a team? How do you manage a team? And so I think right now I don't really have a specific sort of goal. Again, it's more of, hey, this is something that's interesting. Let's pursue it. An extra degree can never hurt, right? And then as I kind of use this degree and my knowledge now to communicate better with customers, understand them better, I think that'll develop over time, right? But unfortunately, right now, it's not necessarily like I have a specific 5-year plan yet. I kind of like just to run with it and then see what the opportunities will open up in the future.
So one last career question. Again, I want to advocate for our guests, our listeners. What is the most for those considering a role like yourself? What would you suggest an academic listener to go for, or to you know emphasize, or what should they be thinking about when trying to make the move to industry and industry positions such as yours?
Yeah, that's a good one. That's a good question. I think, I mean, besides really understanding the science really well, not just in the knowledge aspect of it, like reading papers, that really definitely helps because you have to have that base there. Another one that I think is going to be quite vital for a position like mine is actually having the muscle memory of doing experiments very carefully and precisely, right? And as I mentioned, a lot of times I'm doing demos, troubleshooting, right? So that's where customers done the protocols themselves and didn't get the results they want, right? And so I have to go in there and do the actual experiment with modifications, and then try to get it to work. And a lot of times that would mean you have to be very careful of how you're holding pipettes, how you sort of make sure you pipette the right volumes, how be consistent in mixing the samples or mixing the cocktails of reagents, right? And so that comes with time of having a habit to doing all these small little things in increments. That can really add up to a good isolation, becoming even better, right? So I think if you're in the lab doing experiments, it really helps, I think, to try to practice the best techniques you can. And I know I hated it in grad school, but my PI was always said you have to be as consistent as you can. And I think it really paid off quite a bit for me here.
All right. Well, I think we're going to get ready to wrap up here. So we always end with what we call the fun question, or the not quite career question. So But they're all sometimes related to science too. So for you, we're going to go with, if you could answer any scientific question, regardless of the field or your expertise, you could just hit a button and the answer appears on Wikipedia 100% correct. What would it be and why?
Oh, that's a good one. Yeah, so we're talking about regardless of expertise in chosen field, right? So I would say if I could hit a button and answer a question, it'd be, how do we go warp speed? How do we travel faster than life? Besides, faster than the speed of light. Because growing up, I was just a huge Trekkie. Watched Star Trek all the time, and just the idea that the sky's the limit, the space is the limit, that we can go anywhere we want. I mean, I got to betray immunology here, but that'd be my question that I would definitely want to answer for sure.
Easy. Space travel. I mean, that's just your very, very modest question you have there.
Yeah, just very modest. I mean, if I only had one, right, that seems to be a really good one to answer for sure.
Sounds very good.
I love that one. I want space travel too. Warp drive, here we come.
Absolutely.
You boys, you have fun. I'll just be watching you from Earth while you are, warping to some other galaxy. I'm fine right here. I would go to the moon, but not further than that. I think but that would definitely be a fun thing to answer. So thank you. That was very with that at the end. So finishing on that happy note on our going out to space. Thank you so much for joining us today for the Immunology Podcast. Dr. Wayne Juang from STEMCELL Technologies, talking about his position and a little bit of science, a little bit of career. Thank you so much for joining the Immunology Podcast.
Thank you for being here.
Thanks, Jason. Thank you, Brenda.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at Immunopodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.